Hello friends, today we are going to talk about regulation of gene expression in prokaryotes. Regulation of gene expression basically refers to the process by which the expression of a gene is controlled in response to some external or internal signals. Now, in this universe, each and everything has to have proper check and balance, and genes, that is segments of DNA, we very well know they form the basis of life on this earth. The basic principle, the central dogma of molecular biology, as we all know, it states that DNA by transcription forms RNA, and it is this RNA, the messenger RNA, that translates to form proteins. And these proteins, they act as the structural proteins or the enzymes, the functional proteins, and henceforward. So basically, the expression of these nucleotide sequences, the DNA, it has to be as and when required. It is to be regulated. And this regulation, the various control points of these gene expression, they may include rearrangement of the DNA molecule within the nucleus, whether transcription is possible for a particular DNA part or not. Then regulation during the process of transcription itself. Once the RNA is formed, the messenger RNA in particular, it undergoes certain processing. So again, that can be the checkpoint. After that, this messenger RNA formed, it comes out of the nucleus, and within this cytoplasm, it is translated to form a polypeptide, that is the protein. So next point can be this translation, control of this translation, or the stability of the messenger RNA, that is for how long will it be a functional RNA, will it be possible for it to form proteins. And then these proteins, they undergo further modifications, which are the post-translational control points. So for the regulation of gene expression, these can be the various control points. Now we can say that it is a term for the various cellular processes which control the rate and the manner in which the gene expression takes place. It basically regulates the when and where of specific genes. That is, which genes are activated, when are they activated, and under what circumstances. Some of the genes, they are expressed continuously because they code for the basic metabolic functions, the various proteins which are involved in the basic metabolic functions. Then, as we all know, that after the cell division, they undergo differentiation, structural differentiation as well as functional differentiation. So there are certain genes which are expressed as a part of the process of cell differentiation. That is, because of them, cell differentiation takes place. And once the cell is differentiated, it carries over certain specific purpose for which it is differentiated. So there are other genes which get activated in the differentiated cells, and Now, the regulation of gene expression in prokaryotes that we are going to deal with for our today's topic. That was the general account of what is actually the regulation of gene expression. Now, the basic difference between a prokaryotic cell and a eukaryotic cell is the presence of nucleus. In prokaryotes, nucleus is not present. That means the site of transcription and translation, it is the same, the basic cytoplasm. Furthermore, the DNA is not a complex. The genome organization, the DNA organization is not a complex. So that again is simplified. So the basic three mechanisms that may regulate gene expression in prokaryotes, they are the rate at which transcription takes place, the the life cycle of the RNA, that is the RNA turnover time, for how long is that RNA maintained within the cytoplasm, and then the translation process. Basically, these are the three mechanisms, and the much studied or well known mechanism of the three is the mechanism wherein the rate of gene transcription alters and regulates the expression of genes. Now, in prokaryotes, as we have just said, the genome organization is quite simple. So, here structural units are there, polycistrons are there, that is, all the genes that are involved in a particular metabolic cycle, they are encoded for or transcripted together, and this structural unit is known as a polycistron. Now, when they get activated together, that means they are going to have a common regulatory mechanism, and this structure that results from the combination of the various structural genes of a polycistron and the regulatory sequences, the sequences which regulate the expression of these structural genes, together they form the operon. And this concept of operon was given by Jacob and Monod. So, now there are two types of operons. The inducible operons, that is, which are by default off. Now, one thing I will like to say before this is that the metabolic processes, they can be categorized into two categories. The anabolic processes, that means that they are responsible for the formation of complex molecules from simple molecules, and the catabolic processes, in which complex organic molecules, they break down to form simple molecules. Depending upon the type of metabolic process, these operons, they are also of two types. The catabolic processes, in which a molecule, a complex molecule, is broken down to form simple ones, is known as inducible operon, because it is induced by the presence of the molecule which is to be broken down. So by default it remains off. Lac Operon is an example that we will deal with today. Now, the other type of operon is the one that is related to the anabolic processes. That means the cell, it requires certain complex molecules regularly. So by default, these repressible operons, they are on. They keep on transcribing the messenger RNA on the polycistronic unit until and unless the cell signals that sufficient amount of the product is available, so now it ought to be turned off. A common example of this type of operon is the Operon, which synthesizes the amino acid tryptophan. Now today we are going to talk about the Lac Operon. If we go in by the structure of this Lac Operon has got three structural genes in the form of a polycistronic unit, which codes for enzymes required by the bacterium for the lactose catabolism. These are the lactose permease, which transport lactose into the cell, then the beta galactosidase, which hydrolyzes lactose into the component sugars, that is the glucose and galactose. And the third enzyme is the beta galactoside transacetylase, which is also involved in the hydrolysis of lactose. So these three, the genes for these three enzymes, they are present in the form of a polycistron. Now just upstream, that is towards this 3' end of the Lac Operon, is a sequence, the operator sequence. It is this operator sequence to which certain regulatory proteins, they may be activators or enhancers, or they may be silencers, the repressors, may bind. This is the nucleotide sequence on which these regulatory proteins, they can bind. Now over here at the -10 position is the data box or the Brinbo box, and at the -35 position is again a consensus sequence, and this consensus sequence together forms the promoter region. It is this promoter region where the RNA polymerase binds itself and is responsible for the transcription of this polycistronic unit. Now upstream there are many other enzymes, many other nucleotide sequences, and one of the nucleotide sequence upstream the Lac Operon is the cap site, about which we are going to talk about shortly. And again there are certain repressor genes. These repressor genes, they may be present anywhere on the chromosome. They are responsible for producing the repressor protein that will bind to the operator. Now over here we should see one thing, that these regulatory elements for a polycistron, they are of two types. The ones which are regulating the transcription by virtue of their position, that is the cis acting sequences, the promoter and the operator. These are the regulatory elements, and along with them there are certain regulatory sequences which produce a messenger RNA and a protein, and they may be present anywhere in the chromosome. So they are the transacting regulatory sequences. Transacting because it is by not in close proximity with this polycistronic unit. It is the product which is regulating the expression of this polycistron. Now, if we talk about the lactose, that is the Lac Operon in particular, we have said that by default this remains off because it has to be activated only when lactose is available, which is to be broken down. So when no lactose is there, this repressor gene, it forms a protein, and this protein, this repressor, it binds to the operator site, and thus it blocks the RNA polymerase from moving further to carry on this transcription. As soon as lactose is available, this lactose, it binds to this repressor and prevents it from binding on to this operator site. So this repressor is not present on the operator. Now the RNA polymerase which is present on the promoter point will naturally be free or will be permitted to move ahead and carry on the transcription. So in this manner, these genes, they will be transcribed, and the various 3 enzymes, they will be synthesized, resulting in the breakdown of lactose into glucose and galactose. Now this was the condition when lactose was available for the bacterial cell. Another situation may be that the bacterial cell is exposed to both glucose and lactose both at the same time. So now what will happen within this bacterial cell? Because it has got lactose, and the availability of lactose will induce the Lac Operon to operate, while it is also having the product of lactose. That means under such situations, lactose uh, catabolic enzymes, they are not required. First, the cell will prefer using the glucose that is already available. So this is done by another type of mechanism of regulation of gene expression, that is the catabolite repression. Now for this catabolite repression, one thing we should know is that just now we had talked about that the RNA polymerase, it binds to this promoter region. It needs this cap site, that is the catabolite activator protein site. This cap, that is the catabolite activator protein, it is responsible for the production of cyclic AMP. This cyclic AMP, it binds to nucleotide regions that are present upstream this promoter sequence, and in the presence of this cyclic AMP, this RNA polymerase, it binds to this sequence. That means that in the presence of cyclic adenosine monophosphate molecules, this RNA polymerase binds to the promoter site and then carries on the transcription of this polycistronic unit if the repressor protein is not there, that is in the presence of lactose. So under conditions of high glucose, now how this glucose it affects this process? This catabolite activator protein is now also known as the cyclic AMP receptor protein or the CRP. This cyclic AMP is produced by an enzyme adenylate cyclase, and the glucose it suppresses the activity of this enzyme adenylate cyclase, which forms cyclic AMP. So under conditions of high glucose, this cyclic AMP synthesis is subdued, it is downregulated, and so this RNA polymerase is unable to bind to the promoter sequence, because now this cyclic AMP will not bind to the cap site, the CRP site, that is the cyclic AMP receptor proteins, and hence this RNA polymerase will be loosely bound to the promoter region. Since RNA polymerase is not present or is not properly bound on the promoter site, the transcription won't be at the full rate, that is only low transcription of these three genes will be there. However, once the cell consumes the glucose, now it needs more glucose and has got lactose in its environment. So under such circumstances, that is when there is low glucose, the enzyme adenylate cyclase will be activated, will remain active, and will synthesize cyclic AMP. This cyclic AMP it helps to this CAP, that is the catabolite activator protein, to bind with this cap site. And now because of the presence of this cap protein or the CRP protein, this RNA polymerase it will bind to the promoter, and the rate of transcription of this polycistron will be high. So in this manner we have seen that there are two separate mechanisms under two different environmental conditions that regulate the expression of this Lac Operon. One is the Lac Operon, that is the the inducible repressor, and the other is the catabolite repression, that is the catabolic product of this Lac Operon itself regulates the expression of this polycistronic unit. The catabolite is the glucose, because lactose it breaks down into glucose and galactose. So here we have dealt with the two basic mechanisms of gene regulation in prokaryotes. One is the Lac Operon part, the regulation of the Lac Operon, that is the inducible operon, and the other is the catabolite repression mechanism. In the next part, we will deal with other mechanisms of gene regulation in prokaryotes, that is the Operon, which again has got two mechanisms involved. One is the repressible operon, since it is the repressible operon, and the other mechanism is the attenuation. Thanks for patience listening. We'll talk about other mechanisms in part 2 of this regulation of gene expression in prokaryotes. Thank you.